content. Some years ago, I developed what we call a cycle of surgical research, and you can see it on your screen now. And I'll just go through this because we found that research fellows and even the residents find this cycle of value. It involves 16 steps in the investigation of a particular problem through research. And the reason I put this together was that young people often say, well, I don't see how I could possibly get into research because it's just too awesome. There's too much of it. I wouldn't know where to start. I wouldn't know how to do research. This is sort of a guideline as to the 16 steps that you can take, and you can build on this yourself with your own experience. And this is what we call clinician scientist type of research. That is research that's done by a clinician. It can be basic research, and most of my research with animals has been basic research. But it could also apply to clinical investigation. Now, basic scientists work a little differently, and they don't necessarily go through all of these 16 steps. Basic scientists are more concerned about knowledge for the sake of knowledge, although they were always very happy if something they discovered had a clinical application. Whereas those of us who are clinicians who are doing research are usually trying to solve a given problem related to patient care. And so I'm going to go through, if this is agreeable to you, just to go through this. It starts with recognizing an unsolved clinical problem. Now, often the residents will say to me, well, everything's been studied, everything's been investigated through research, surely. Surely there's nothing more to be done. And I said, there's a great deal more to be done. But you have to recognize an unsolved clinical problem. And we use an example for, let's look at congenital dislocation of the hip. There were many forms of treatment for this condition. The results were all very bad, all over the world. So that was an area that needed investigation. But you have to recognize that it needed that, not just go on doing what had been being done before. And as I mentioned to you when I spoke about the McLaughlin Scholarship, one of the things that really impressed me during that year was that so much was being done from day to day in teaching hospitals, good surgeons, on the basis of empiricism. That is to say, they did something just because it worked. Used aspirin for a headache, didn't know how it worked, but it worked, so I did it. And life of a surgeon can be almost entirely empirical, but for an academic surgeon, one has to recognize unsolved problems. And then the first step is to think, to contemplate, to speculate. And this requires time. It's not something you can usually do during the day, during a busy day where you're treating patients, solving problems for residents or colleagues and so on. It requires a quiet time, usually in your study at home, when you really think seriously about the problem and contemplate about it, speculate. And then the next step is to review the scientific literature and look at the historical background. And it was Churchill and others, I think, who said that he who does not read history is doomed to repeat the errors of the past. It's been credited to a number of different people. But it's very relevant to this research cycle, because if one does not read the scientific literature, one is likely to repeat work that has been done before, to repeat experiments that have been done before, even those experiments may have been very negative and so on. So that reviewing the scientific literature is really very important. And in doing so, one often has a clear idea of what you might do that is different. And then the next step is to ask intelligent questions. Why, how, what, which. The sort of questions that a reporter asks when they come on the scene of a crime, or a detective asks. But the questions have to be intelligent questions. And there's some questions, for example, that don't lead themselves to research. If you were to ask the question, which came first, the chicken or the egg, it's not a question that you can answer through research. So they have to be intelligent questions. And Sir Henry Tizard had said the most important part or step in research is to ask an intelligent question. And then the next step, which is right here, is to formulate an hypothesis. This is an interesting word, hypo, meaning under, like hypodermic, or less than, and then less than the final thesis. And it's really just a provisional supposition or possible explanation. Now, you can use the null hypothesis, which is what Sir Karl Popper taught, or you can use just the ordinary hypothesis. It's not important which you do. But you have to develop an hypothesis that you're really going to test. Now, you should never do research to prove that your hypothesis is correct, because then you're unlikely to really get the truth out of your findings. You're likely to be biased. So you should do research to test the validity of the hypothesis. And if your hypothesis proves to be wrong, that's fine, because you've learned something and you develop a different hypothesis. But if you just try to look for those results in your research that favor your hypothesis, then you're going to mislead yourself and mislead other people as well. So the hypothesis is a very important part of the whole story. And then the next step is to plan the research protocol, the strategy of the research, the investigation, including the methods of the study and controls. The control is the most important part of any scientific investigation. You don't just do an experiment with animals, for example, or look at problems with patients without having something to compare it with. So you have to have a control group of animals, sometimes several types of control to compare it with. Otherwise, you're just working in a vacuum. And then the next step in this cycle is to seek collaboration, that is interdisciplinary research. In England, I used to see what's called a one-man band. You see some fellow on a street corner with a ukulele and a brass horn and a drum and several other instruments all playing at the same time. And it didn't sound like a symphonic orchestra at all, but this was a one-man band. Now, there's no place in research anymore for the one-man band. When I started research in 1954, while I was still in England, I could do my research sitting at a microscope with one research fellow and perhaps ask from time to time other colleagues what they might think. But now, it's very different. We do collaborative research that's multidisciplinary. And I work with physiologists, with pathologists, geneticists, for example, biomedical engineers, and all kinds of individuals who are in different fields altogether, but work together as a team. And then the next step in this cycle is to apply for funding. And young scientists find that the most difficult grant to obtain is the first one. And those of us who are senior should always be willing to help the young people with their first grants, so that there is something called grantsmanship, which is important, and which they have to learn. And there is still money available for good research. But you have to be able to state what you're planning to do very clearly. You can't expect a review committee to just try and figure out what you're aiming at. You have to be very, very clear in how you apply for it. And that's the first time that you're involved with peer review. And then you conduct the investigation. Now, because of our belief in the sanctity of human life, we would never do experiments with human beings. We do clinical studies, the results of treatment and so on, but not experiments. So we do experimental work with animals and the clinical studies with patients, but always remembering the sanctity of human life. And then we're coming around on the left-hand side of this cycle, coming back up toward the original problem. We collect and analyze the data. We look at the statistical significance. And the epidemiologists have become very sophisticated in looking at evidence. So that, this was started in this country largely by Dr. David Sackett, but there are many people now, including our current dean, Dr. David Naylor, who are superb epidemiologists and know all about statistics. So what you're doing has to be analyzed by a statistician. And at this site, at this particular time in the cycle, we come across from time to time the phenomenon known as serendipity. Now, that's a lovely word, serendipity. It was coined in, I think, the 15th century by Hugh Walpole. And it goes back to the time when Sri Lanka was known as Serendip. And then it became known as Ceylon and now Sri Lanka, but it's the same country. And there is a story, it was mythical of course, that there were 3 princes of Serendip who set out on a long journey, and they had a specific goal in mind, which they never did reach. But on the way, just by chance, they came across something that was even more important than that which they had been seeking. And so Walpole coined the term serendipity. And an example of this is, for example, the discovery of penicillin. And Sir Alexander Fleming was a rather untidy scientist, and he went away for a weekend leaving some petri dishes in which he was growing staphylococci. He left the tops off them. And when he came back at the end of the weekend, he noticed that there was a mold had contaminated some of these petri dishes, and wherever the mold was, there were no bacteria. And he analyzed that mold, and it was Penicillium notatum. I have a slide from a cartoon I often show the medical students, and it shows two sweeping ladies in a laboratory, and Fleming's name is on the laboratory, and one is saying to the other, if that old man thinks that I'm going to tidy up his dirty old dishes, he has another thing coming. So it's a good thing they didn't tidy up the old dishes. But that is an example of serendipity, and there have been many other examples through the ages. And indeed, you have to keep your eyes open for that, because you may stumble over it. Churchill once said most people, when they stumble across the truth, manage to pick themselves up and carry on as though nothing had happened. Well, if you're really alert, you sense this phenomenon of serendipity, and that, in a sense, can start a whole new cycle. And this is why a good scientist never really runs out of ideas, because he keeps finding things that are really important, and starts a new cycle. And then you have to interpret the data. And here's an important concept, is that you have to make the theory fit the facts. Now, some scientists have a wonderful theory in their mind, and they try to make the facts fit the theory, but that's the wrong way to do. You have to make the theory fit the facts. And then you have to draw valid conclusions based on factual data and logic and on reasoning. I mean, an example of lack of logic would be when coffee is being served, the plane reaches some turbulence, and they say, well, it's because coffee was being served that it became turbulent. And there are many examples of inappropriate conclusions being drawn. So then you get to the point halfway up toward the original problem again, where you're answering the original questions. And then you have to present that at a meeting, scientific discussion of other people, of your peers. This is the second place where peer review comes into importance. And if you're going to be in the big leagues, just as in sport, if you're going to be in the big league, you have to be a big leaguer. And you have to be willing to accept criticism. Now, some scientists are so precious and so thin-skinned that they get upset by criticism. I've always felt by criticism that I learned something, and that as you go through life, you should have your eyes going straight ahead and not look to the left for the applause or to the right for the criticism, because otherwise you're just like watching a tennis game. You have a horizontal nystagmus going back and forth. But to just keep your eye on the ball the whole time. Listen to the criticism, learn from it, and don't be swayed too much by the applause or the accolades on the other side. And then, having presented the work for discussion, having learned more from that, you then have to publish it, because if you don't publish it, it's lost when you go. And this is another place where the peer review comes in. And as I said earlier, I think that it's unfortunate that we've developed this sort of publish or perish, because it just means too much is being written that isn't really all that appropriate. I have another cartoon that shows two men at a gravestone. They're saying, well, poor old Atkinson, he published and published and he still died. So publish or perish is not a very good way of looking at it. And then we apply the new knowledge back to the original problem and improve that situation, either better diagnosis or better prevention or better treatment or better understanding of the disease. So those are the 16 steps of the cycle of research. And put in the center here, one good research cycle begets another, particularly on this left side, up here where we're coming back up, and you see something that you didn't expect, and that will set up a whole new research cycle.
It's interesting that you take us so carefully through that. That's a kind of tour de force. And I guess, as you said at the beginning, it's not something that comes naturally to young students.
It requires quite a bit of discipline. unless a scientist is really disciplined to follow the rules of science, which are really very, very important and very strict, particularly with having controls, for example. But unless a scientist is willing to do that, then he's not going to succeed.
And you mentioned grantsmanship, and there's a certain amount of, I guess, gamesmanship in this?
I wouldn't say gamesmanship so much as being an educated way of approaching sharing your plans with a review committee. And there are certain ways of doing this. And gamesmanship sounds almost as though there's an element of deception, but this is not deception. This is grantsmanship, being able to write clearly. Well, it's a bit like writing ability and writing a textbook and that sort of thing. But it's a very important part of it. And one must never be discouraged about having a research project turned down, particularly in the early part of your life. But unfortunately, quite a few budding scientists who become academic surgeons soon find that it's so difficult to get money for their grants, and it's such a demanding field that they soon allow the patient care to crowd in on their life, and the research gets dropped. And then that starts a downhill cycle, because the less research they do, the less they have to report for the next grant application, the less likely they are to have it approved. And then they start saying, well, the system isn't fair, you see.
And this is the problem that clinicians often complain about, that they're second-class citizens when it comes to research and funding for research.
Well, I think this is...